Beneath the sands of Libya, scientists unearthed two mummies with DNA that shouldn't exist. Their genetic code matched no one alive today. No sub-Saharan African, no Egyptian, no Mediterranean population, no modern human group anywhere on Earth. For decades, geneticists had only glimpsed faint echoes of this mysterious lineage in the genetic background of modern humans, whispers in the genome, evidence of ghosts, people who had vanished long ago without leaving physical traces. But in 2025, researchers finally dug up the source. They uncovered the actual skeletal remains, the mummified flesh, the preserved genetic material itself. What they discovered rewrites everything we thought we knew about human migration, ancient Africa, population genetics, and the peoples that simply vanished from history without explanation. The story begins in 2003, when archaeologists working in the Tadrart Akakis Mountains of southwestern Libya stumbled upon the Takar Kori rock shelter. It wasn't a dramatic find at first, just another ancient site buried beneath layers of desert sand and accumulated time. The location itself seemed unremarkable, a rocky outcrop in one of the world's most inhospitable regions. But over the next 3 years, between 2003 and 2006, the excavation team working methodically through the shelter uncovered something extraordinary. Buried in the shelter were the remains of 15 individuals dating back thousands of years. Most were women and children, their bones and artifacts scattered across multiple occupation layers. But 2 of them had undergone something rare in archaeological contexts, something that would preserve their genetic secrets intact for 7,000 years. They had naturally mummified, their bodies preserved by the unique environmental conditions of the rock shelter. The Sahara is the world's largest hot desert, spanning nearly 9 million square kilometers across North Africa. Its surface temperature can soar beyond 130 degrees Fahrenheit during the day, then drop dramatically at night. These extreme thermal oscillations are devastating to biological material. DNA, the fragile molecule that encodes genetic information, typically doesn't survive in such hostile conditions. The heat accelerates. Chemical degradation. The aridity desiccates tissues, but also exposes DNA to oxidative damage. The relentless passage of millennia, 7,000 years of exposure to these elements, should reduce genetic material to useless molecular fragments. Scientists have long struggled to extract ancient DNA from African archaeological sites for this exact reason. The continent's hot, humid climates in many regions, combined with its desert's extreme temperatures, make DNA preservation rare. Most ancient African remains yield little or no genetic information. The Takar Kori mummies defied this expectation entirely. The combination of the rock shelter's specific microclimate, protected from direct sunlight and extreme temperature fluctuations, the naturally desiccating conditions inside, and perhaps some fortunate circumstances of how the bodies were positioned and buried, had done this supposedly impossible. They had preserved these women's genetic code intact enough to sequence completely, a rarity that would excite any paleogeneticist. For years, the Takar Kori remains sat in laboratories while researchers analyzed them using older techniques. Earlier studies focused on mitochondrial DNA, the genetic material passed down through the maternal line. It's limited, containing only about 16,000 base pairs compared to the 3.2 billion base pairs in a full human genome. But mitochondrial DNA is tougher, more resilient. It survives where nuclear DNA fails. The initial results were puzzling. The women's mitochondrial DNA didn't match expected populations. But without access to their complete genomes, scientists couldn't tell the full story. That changed in 2024 and 2025, when researchers at the Max Planck Institute for Evolutionary Anthropology, led by archaeogeneticist Neda Salem, successfully sequenced the entire genomes of both individuals. Johannes Krause, the senior author and director of the Department of Archaeogenetics at Max Planck, described the moment as remarkable. In hot climates, DNA breaks down into tiny fragments. Reconstruction requires not just fragments, but enough of them to stitch together the complete picture. The Takar Kori women were among the oldest natural mummies ever to have their full genomes recovered. That achievement alone was groundbreaking, but what those genomes revealed was something else entirely. When Salem's team analyzed the results, comparing the Takar Kori women's genomes against databases of modern and ancient human DNA from around the world, they found something that contradicted every assumption about North African population history that researchers had held for decades. The women's genetic makeup was supposed to reflect a simple, elegant story. Humans migrated out of Africa into the Middle East, Europe, and Asia. Some humans later migrated back into Africa from these regions, bringing new genetic variants with them. Populations mixed and mingled across geographic boundaries. Genetic diversity in this model tells the story of constant movement and connection, of populations colliding and combining to produce the modern human genetic landscape. But the Takar Kori women told a completely different tale. Their DNA indicated they belonged to a human population that had split off from sub-Saharan Africans approximately 50,000 years ago. Not 30,000 years, not 20,000 years. 50,000 years ago, back at the exact time when other human groups were first beginning to leave Africa entirely, to migrate into Europe, Asia, and beyond into the first great diaspora of our species. After that initial split, the Tucker-Corey lineage never mixed with outside populations. For 50,000 years, 500 centuries, this population existed in complete genetic isolation. Think about that timescale. 50,000 years encompasses the entire modern human occupation of Europe and Asia. 50,000 years saw the development of agriculture in the Fertile Crescent, the rise of civilizations in Mesopotamia and Egypt, the construction of pyramids, the forging of empires. Throughout all of this time, the Tucker-Corey population didn't interbreed with populations moving north from sub-Saharan Africa, despite living in the same general geographic region. They didn't mix with Mediterranean peoples across what would become known as the sea. They didn't absorb genetic material from Levantine or Middle Eastern populations with whom they presumably had contact. For 100,000 years, they remained genetically pure, locked in their own separate branch of the human family tree, maintaining reproductive boundaries with every other human population. That isolation persisted all the way until their deaths around 7,000 years ago. The women themselves lived during what scientists call the African humid period, a climatic interval, when the Sahara was not the barren wasteland we know today, but something entirely different. Between roughly 14,800 and 5,500 years ago, North Africa experienced something extraordinary on a continental scale. Climate patterns shifted dramatically. Monsoon rains that had previously turned back at the coast penetrated deep inland. Rivers flowed where dunes now stretch endlessly for hundreds of kilometers. Lakes filled depressions in the landscape, creating islands of water in what would later become dry land. Grasses grew in vast prairies. Trees flourished in quantities that seem impossible in our modern understanding of the Sahara. The Sahara transformed into what researchers now call the Green Sahara, or the Humid Sahara, a verdant savanna landscape teeming with life from species that would take decades to encounter there today. Hippopotamuses wallowed in rivers, their massive bodies cooling in waters fed by reliable rainfall and underground aquifers. Giraffes browsed on acacia trees across the grasslands. Wild cattle roamed freely across savannas. Elephants moved through forests. Lions hunted, and humans thrived in this transformed landscape. The Tucker-Corey rock shelter was one such human settlement within this green Sahara world. The two mummified women lived during this period of climatic abundance and environmental richness, perhaps 2,000 to 3,000 years after the Green Sahara began forming from the previous arid conditions. They were pastoralists, herders who tended domesticated cattle and other livestock, practicing an economy that relied on intimate knowledge of seasonal water availability and grassland productivity. The excavation at Tucker-Corey, conducted between 2003 and 2006, revealed pottery decorated with designs that reflected pastoral lifeways, stone tools suited to herding and food processing, and abundant animal bones that testified to this pastoral lifestyle based on livestock management. The women would have lived in a world of open grasslands stretching to the horizon, reliable freshwater sources from lakes and rivers, and relatively comfortable climatic conditions. They would have known the Sahara not as a hostile desert threatening death and suffering, but as a homeland, a region of resources and opportunity. But here's where the mystery deepens. The Green Sahara, despite its favorable conditions, did not act as a migration corridor. Archaeologists had long theorized that the humid period would have encouraged movement and mixing between sub-Saharan, African, and North African populations. The climate was perfect for travel. Resources were abundant. Populations should have been mobile, trading, communicating, interbreeding. The conventional model predicted genetic mixing on a massive scale. The Tucker-Corey women's genomes said otherwise. Approximately 93% of their ancestry derived from a previously unknown North African population that remained isolated. This was not a population that participated in the movements and migrations reshaping the rest of Africa. This was not a people who received genetic input from southern neighbors. They were locked in their own world, shaped by their own evolutionary pressures, developing their own genetic uniqueness. The researchers who published these findings in the journal Nature in late 2025 found themselves forced to reconsider fundamental assumptions about African history. Genetic isolation on this scale, maintained over 50,000 years, was unusual. It suggested something deeper, perhaps geographic barriers, perhaps cultural preferences for endogamy, perhaps a combination of factors that kept populations apart. The women's genomes revealed another intriguing detail. They carried traces of Neanderthal DNA. This finding was itself unusual. Neanderthals were European and Western Asian populations that went extinct approximately 40,000 years ago. They never lived in Africa. Any Neanderthal DNA in modern human genomes entered through interbreeding between African-descended humans and Neanderthals outside Africa, typically in the Near East. The Tucker-Corey women's genomes contained approximately 0.15% Neanderthal DNA. That's incredibly low, roughly a tenth of the amount found in Europeans and Asians today. It's even lower than most ancient or modern sub-Saharan Africans. Yet the presence of any Neanderthal DNA was significant. It meant that, at some point, this isolated North African population had limited contact with populations connected to the broader Near Eastern network, perhaps through rare trade encounters, perhaps through occasional migrations along coastal routes, perhaps through movements along the Mediterranean fringe. The signal was faint, but it was there. The Tucker-Corey people were not completely sealed off from the outside world. They simply chose to remain genetically distinct, a population whose cultural networks extended further than their reproductive networks. This distinction between cultural and genetic isolation became central to understanding these women and their world. The excavations at Tucker-Corey revealed pottery and artifacts that clearly came from sub-Saharan Africa. The archaeological record shows that cultural exchange was robust and continuous. Ideas moved, materials moved, trade goods moved, artistic styles moved. Everything except genes seemed to move freely across the supposedly isolated Saharan landscape. Savino di Lernia, an archaeologist from Sapienza, University of Rome, who directed excavations at Tucker-Corey, reflected on this paradox. The Tucker-Corey people were isolated genetically, he explained, but not culturally. Networks connected them to distant regions, but these networks did not involve reproduction with outside groups. This tells us something profound about human behavior in ancient times. Cultural contact does not require genetic mixing. Peoples can trade, share ideas, adopt innovations, and remain socially separate. The pastoral tradition that the Tucker-Corey women practiced, herding cattle and other livestock, was not invented by North Africans. It spread from the Near East, from populations that had developed domestication and herding thousands of years before. Yet, when this technology reached the Green Sahara, it was adopted, adapted, and refined by people who did not intermix with its originators. Knowledge crossed boundaries that genes did not. The question of how and why this isolation maintained itself for 50,000 years invites speculation. Geography likely played a role. The early Green Sahara period may have been bounded by different ecological zones that limited movement. The later Green Sahara, by the time the Tucker-Corey women lived, existed within a broader Saharan geography that presented its own barriers. Mountain ranges, arid regions, and shifting resource availability could have constrained long-distance movement. But geography alone doesn't explain 50,000 years of complete reproductive isolation. Cultural practices must have reinforced the genetic separation. Perhaps marriages with outsiders were forbidden. Perhaps outsiders were simply rare enough that such pairings never occurred. The climate records tell us what happened after the Tucker-Corey women died. Roughly 5,500 years ago, the African humid period ended. The monsoons shifted. The rains stopped penetrating inland. Rivers dried. Lakes evaporated. The Green Sahara contracted. Grasses gave way to sand. The comfortable pastoral world transformed into harsh desert. Populations were squeezed into smaller habitable zones, along coasts, in mountain refugia, in the remaining river valleys. The Tucker-Corey rock shelter itself became increasingly marginal. Archaeological evidence suggests the site was abandoned around this time. The pastoral communities that had flourished there either migrated elsewhere or were absorbed by neighboring populations. But the Tucker-Corey lineage appears to have disappeared. Their genetic signature vanishes from the archaeological record after the climate shifted. For thousands of years afterward, through the Bronze Age and Iron Age and into the Classical period, no genetically pure descendants of the original Tucker-Corey population appear in the archaeological samples. The lineage that maintained itself in isolation for 50,000 years was erased in a few centuries of climate disruption. This speaks to the fragility of small, isolated populations when environmental conditions change suddenly. They have no genetic diversity to draw upon. They have no adaptive innovations from neighboring populations flowing in. They are locked into their particular evolutionary trajectory. Yet the Tucker-Corey people were not completely forgotten by human genetics. Faint echoes of their lineage persist in modern North African populations. The genetic signal is ghostly, barely visible through the noise of 20,000 years of subsequent mixing and migration. But it's there, like the faintest echo in a canyon. A whisper that someone once lived here, someone whose genetic legacy was absorbed and diluted by newcomers, but never entirely erased. This is where the popular headline about mummies, whose DNA doesn't match any living human, becomes misleading. The genetic material of the Tucker-Corey women survives in modern North Africans. The connection is attenuated, diluted by millennia of subsequent mixture, but it exists. The genome analysis also revealed connections to other ancient North African populations. The Tucker-Corey women shared genetic links with individuals from Tafralt Cave in Morocco, dated to around 15,000 years ago. and associated with the Ibero-Mauritian culture. These were hunter-gatherers from an earlier time, before the Green Sahara period. The connection suggests that a stable human population existed in North Africa across the transition from late Pleistocene to early Holocene, persisting through the humid period and beyond. The Takakori lineage may represent a continuation or related branch of this deep North African population history. Nada Salem, the lead researcher, emphasized that these findings challenge previous assumptions about North African population history. For decades, researchers operated under models that emphasized migration and admixture. The idea was that Africa, like the rest of the world, was constantly being reshaped by population movements. Groups would arrive, interact with earlier populations, and contribute their genes to the next generation. Genetic diversity reflected this narrative of constant mixing. But the Takakori women's genomes suggest a different pattern, one where populations could remain completely separate for vast stretches of time, despite living in the same general region and engaging in cultural exchange. This has implications for how we understand human population structure more broadly. In Europe, by contrast, ancient DNA has revealed much more genetic admixture and replacement. Different populations mixed continuously. Invading groups left genetic signatures in resident populations. The pattern of migration and mixing seems to have been qualitatively different between Africa and Europe during the Holocene. Why? Perhaps African geography, with its vast, diverse landscapes, allowed populations to remain geographically separate more easily. Perhaps African cultural practices more strongly enforced reproductive boundaries. Perhaps the answer involves a combination of environmental and cultural factors that ancient genomes alone cannot reveal. The practical challenge of extracting DNA from the Takakori mummies deserves emphasis. These women were buried in a hot desert 7,000 years ago. They should not exist in terms of DNA preservation, yet they do. The luck of burial conditions, a rock shelter providing protection, desiccation preserving tissue, stable temperatures, allowing natural mummification, allowed these genomes to survive. Researchers extracted thousands of DNA fragments from teeth and leg bones. Modern sequencing technology, capable of reading degraded fragments, made reconstruction possible. This is why the Takakori mummies matter so much beyond their genetic story. They represent a proof of concept. DNA can survive in Africa's hot deserts under the right conditions. This opens possibilities for future discoveries. The genomes are now helping archaeologists reframe questions about the Green Sahara period. Why did pastoral economies develop in the Sahara? Was it through population replacement, with outsiders bringing the technology of herding? Or was it through cultural diffusion, with local populations adopting and adapting the technology themselves? The Takakori women's genomes suggest the latter. A local North African population adopted pastoral practices invented elsewhere, but maintained genetic separation from the populations that originated those practices. This challenges models of technological spread based on migration and replacement. The discovery also speaks to what archaeologists call the ghost populations, or ghost lineages, that existed in human prehistory. For decades, geneticists noticed faint genetic signals in modern humans that suggested admixture with populations that had vanished without leaving clear traces in the archaeological record. These echoes appeared as subtle statistical signals in genome-wide association studies, but had no physical remains to attach them to. The Takakori women's skeletal remains, preserved and now genetically characterized, represent one instance where a ghost lineage was finally found in flesh and bone. This validates the idea that populations leave genetic fingerprints on their descendants, even when their archaeological record is sparse or absent. The study was published by an international team of researchers from multiple institutions. Beyond Salem and Krauss at Max Planck, contributors included David Caramelli from the University of Florence, Savino di Lernia from Sapienza University, and many other archaeologists, geneticists, and specialists. The collaboration represents the modern approach to archaeogenetics, combining ancient DNA expertise, archaeological knowledge, dating techniques, and statistical analysis to extract maximum meaning from limited remains. No single discipline could have told this story alone. The interplay between genetics, archaeology, and paleoclimatology was essential. The media coverage of these discoveries illustrates why scientific literacy matters. Viral headlines declaring that scientists found mummies with DNA matching no living human create the impression of something impossible or alien. The sensationalism invites misunderstanding. People imagine DNA that doesn't belong to humans at all, or this was a population at a dead end. Their isolation, which made them genetically pure and distinctive, also meant they lacked resilience. When climate changed, when the Green Sahara contracted, they had no genetic flexibility to draw upon. They couldn't hybridize with incoming populations to create offspring with new adaptive combinations. They couldn't move en masse and successfully compete for resources in new environments against populations with different genetic profiles. They couldn't weather the transition from a pastoral paradise to a harsh desert. They were locked into their particular genetic and cultural package. And when the world changed, that package became obsolete. Yet their story is not one of failure or tragedy, at least not when viewed from the perspective of human history writ large. The Takakori women lived for 7,000 years in a time and place of relative abundance. They practiced a successful pastoral lifestyle. They participated in trade networks and cultural exchanges. And they maintained their traditions and their genetic identity across millennia. They were not primitive or backward. They were simply people who chose to remain separate, genetically, from their neighbors, and who flourished under conditions that allowed that separation. When those conditions ended, they ended, but they had lived fully and completely for thousands of years. This is what ancient DNA allows us to do: resurrect the voices of the voiceless, give shape to the shadows of prehistory, confirm the existence of populations that left only whispers in later generations' genomes. The Takakori women died 7,000 years ago, yet their genetic legacy speaks across the millennia to modern researchers. It tells of a world far different from the Sahara we know. It tells of populations making choices about whom to marry and whom to avoid. It tells of cultural networks spanning vast distances alongside genetic boundaries between peoples. It tells of climate change, erasing carefully maintained systems of living. And it tells us that human history is far more complicated than simple narratives of constant mixing and migration can accommodate. The implications extend beyond North Africa. If populations in the Sahara could maintain 50,000 years of genetic isolation, despite favorable climatic conditions and evidence of cultural exchange, what other isolated populations might have existed elsewhere? How many ghost lineages survive in the genetic background of modern humans? Traces of peoples who achieved local stability and then vanished? How many branches of the human family tree are we yet to discover through ancient DNA, branches that will force us to redraw our models of migration, adaptation, and cultural development? The Takakori mummies are part of a broader revolution in how we understand human prehistory. Each new ancient genome adds pieces to a puzzle that's far larger and more intricate than previous generations imagined. The story of human history is not a simple narrative of expansion and replacement. It's a complex mosaic of populations, each with their own trajectory, their own choices, their own unique combination of genetic inheritance and cultural innovation. Some populations mixed freely, some remained isolated, some had advantages that allowed them to spread widely. Others maintained tight boundaries and thrived locally. All of these patterns, when traced through ancient DNA and archaeological evidence, reveal a human past of remarkable diversity. Nadasalem's team plans to continue research in the region. More samples from the Takakori site and related locations could reveal whether the genetic isolation was unique to this particular group or representative of broader patterns in the Green Sahara. Understanding why this population chose or was forced to remain reproductively separate requires continued investigation. The environmental conditions that permitted isolation need to be mapped and understood. The archaeological record needs to be excavated more thoroughly, and new technologies in DNA analysis may reveal additional details about these women's genetics, their diet, their migrations, their relationships. The discovery also highlights why African archaeogenetics remains a frontier. The continent's diversity of climates, populations, and histories has barely been scratched by genetic investigation. For every Takakori mummy that survives, countless remains have been lost to time's erosion. The gaps in our knowledge of African prehistory are vast. New discoveries will continue to surprise us, to contradict our models, to reveal populations and patterns we never anticipated. Each genome sequenced from an African archaeological site has the potential to reshape our understanding of human history. The popular fascination with sensational headlines about mummies and mysterious DNA reflects a genuine hunger to understand our own origins. Where did we come from? How did different populations emerge? What separated us into the diverse peoples of the world? These are fundamental human questions. The Takakori women's genomes provide real answers to these questions. They show us that human populations can differ genetically in profound ways while remaining fully human, fully capable, fully worthy of study and respect. They demonstrate that genetic variation is not random, but reflects historical choices and circumstances. They remind us that behind every genetic signal lies a story of real people living real lives. The Takakori discovery ultimately tells us something about the nature of science itself. Sensational headlines capture attention, but they often distort the actual findings. The real story, a population that maintained genetic isolation for 50,000 years, that engaged in cultural exchange despite reproductive boundaries, that thrived and then vanished when climate changed, is more interesting than the clickbait version. It's more human, more nuanced, more reflective of the actual complexity of life and history. This is why understanding the difference between headlines and science matters. The genuine discoveries of archaeogenetics are already remarkable enough without sensationalism.